My name is Laurel Heetland. I go to Lancaster High School as a ninth grader, and this year I chose to do my project on coliform bacteria. I chose this project because I want to go into animal conservation science in the future, and a lot of that is environment and water and animals and plants, and I figured this would be a good step in that direction. When researching my background information, I found that coliform bacteria is a rod-shaped bacterium found in the intestinal tracts of human and other animals. It can cause stomachaches, diarrhea, and other gastrointestinal diseases if ingested through the mouth, nose, ears, or eyes. It is referred to as an indicator organism because they indicate the potential presence of disease-causing bacteria in water. People can become immune to coliform bacteria if they're used to drinking it throughout their life in small amounts. But if others who are not immune to it drink the same water, they risk getting sick. Natural water sources are more likely to contain coliform bacteria because of animal waste and road runoff, which is also a part of my hypothesis that natural water sources are more likely to contain coliform bacteria. It is more common for drinking water in Sudan or any impoverished country to contain coliform bacteria because they lack the proper filtration systems. An example of this again is Sudan, because most of their drinking water gathered is from natural water sources, which in my data was proved that they're more likely to contain coliform bacteria, and they don't have the money needed to access filtration systems. How I tested the water was 1 milliliter of water was measured in a pipette and added to a test tube containing violet red bile agar and left to sit for 48 hours. After 48 hours, if the test was still negative, which showed a reddish tint, the test was left for another 24 hours. After the incubation period was over, if the test still showed a red tint, which was negative, or if it showed a yellowish tint, it was positive. On average, the positive tests were yellow after about 24 hours. After all the samples were tested twice and the 78-hour incubation period was over, the data collected was recorded in multiple graphs and tables. For this, I needed a coliform bacteria test kit, which contained 30 test tubes with violet red bile agar, and violet red bile agar is essentially the inner lining of a cow's stomach that the bacteria uses to feed off of, having a chemical reaction with it and changing the color if it is present and if it's not. I used rubber gloves, which were provided by my teacher, the pipette to measure the water samples, and then rubbing alcohol to sterilize everything. How I sterilized it was I flushed everything out with the rubbing alcohol after the test was conducted, and before the next test, I rinsed it all off with the water that was going to be next tested. So the rubbing alcohol would be diminished, but also so the bacteria wouldn't be killed, so I had an accurate result. My results show that the mode of various water tested for coliform bacteria was negative for well water and city water, but was positive for natural water. The frequency distribution of city water was 1 negative, and city water was also my control because the water is tested and filtered thoroughly. Well water had a range of 3 positives and 8 negatives. The 3 natural water samples collected, 2 of them were positive and 1 was negative. I expected most of the natural water tested would be positive because of animal waste in the water. I assumed little to no coliform bacteria would be found in well water, but was surprised with just how much that was present. After the city water was tested, there was no coliform bacteria, which I expected because the water is treated thoroughly and filtered and was used as my control. This table shows the mode of the varying water sources was negative for city water, negative for well water, and positive for natural water. The frequency distribution was 0 for city water and 1 negative for city water, 3 positives for well water and 8 negatives for well water, and 2 positives for natural water and 1 negative. The number that each sample was tested was twice for every category, and the percent of positive test results containing coliform bacteria was 0% for city water, 27% for well water, and 67% for natural water, which is by far the highest amount of present coliform bacteria. This graph shows that well water out of the 11 tested had 8 negatives and 3 positives. The 1 city water, or the control that was tested, was 1 negative, and the 3 natural water sources tested, 2 of which were positive and 1 was negative. This is my appendix of raw data. As I collected the data, it was essential that I kept it organized in a table because each one was tested at a different time and date. So in order to keep track of the incubation period, I put it in a table with each sample and the location, time, and date, and then the results. If I were to do this project again or further expand on it next year, I would consider using distilled water or boiled water after a control rather than city water. And just because there's still a potential possibility that there would be coliform bacteria in the city water, even though it's tested and thoroughly. I would consider testing more water samples and expand the location of samples. So I only tested water from Fairfield County, but I would like to have tested more samples from outside of Fairfield County. And I believe it would benefit the test if each sample was tested more than twice to ensure absolute correct answer. So I tested each sample twice, but I think 3 or 4 times would have been beneficial to rule out any possible human error or problem that went wrong during it. And I would have liked to have been able to see the bacteria for myself under a microscope to further expand my knowledge on it and help with background knowledge and be able to inform people of it more. The people that did have positive water sources were informed and ensured that they shocked their water. To do this, they used liquid laundry bleach to shock chlorinate the water system. 2 quarts of bleach is added to 10 gallons of water. The bleach was poured down the well and was circulated through the entire water distribution system. It was then left for 6 to 12 hours, and then for the chlorine to start working, and then flushed thoroughly with water through the well and the pipes. Thank you for watching my project.